Thanks for being here, everyone. I'm Ryan. As Eric said, this is accelerating single-cell bioinformatics with n-dimensional arrays in the cloud, which is a lot, but we're going to get through it in 3 parts. Single-cell sequencing is an exciting new measurement technology with profound implications for a lot of biology and medicine. It generates array data that we want to store and process. And this being 2019, we often want to do that in public clouds or various cluster contexts. And that presents interesting challenges. So I work at Mount Sinai in New York at Uri Laserson's lab. And this work was funded by the Chan Zuckerberg Initiative and is part of the Human Cell Atlas, which is a big consortium essentially dedicated to standardizing around these single-cell analyses, how we perform them, what the data should look like, what the tools that process that data should look like, et cetera. And my colleague, Tom White, in particular, did most of the work I'm going to talk about today. There's a slides bit.ly that's at the bottom of the slides. Maybe hard to see. It's bit.ly PyData-RW. Lots of things here are linked if you want to follow up there. So, agenda, basically, I'll talk about why single-cell sequencing is interesting and exciting. Essentially, it allows us to go from bulk sequencing of cells to resolving the RNA of individual cells, which allows us to just know many more things than conventional genome sequencing has allowed. Storing the data, today, most of the tools that operate on this data use HDF5 as an archival storage format. And there are some rough edges to using it. And we've been somewhat evangelizing a upstart container storage technology called Zarr that's very similar, but plays more nicely with parallel compute paradigms and cloud storage. So I will talk about the 2 of those and other relevant technologies in the space. And I'll talk about work we've done to allow existing tools that already process this data to be run seamlessly in parallel using a variety of parallelization backends that we're living through a small renaissance in. And there are many interesting choices that you may like to try out. So a quick, somewhat silly exercise is we'll just contextualize some of the basic biology that's relevant to understanding why single-cell sequencing is interesting. The biological information essentially progresses from DNA to RNA to proteins, which is analogous to information on your computer being stored archivally on a hard disk. It's very large in size. You're typically not using most of what you're storing at any given point. The bits that you do want to use, you page into memory, which is like RNA. It's more volatile and ephemeral, much smaller in size, though the information is still very identifiably the same as it was on disk, just in a different physical substrate. And eventually, all of the work actually happens by proteins interacting and essentially performing computations by their 3D conformations binding to one another or not. And that's kind of like how the information gets chopped and massaged in various ways and ends up in the CPU registers and actually goes out and affects the world. Maybe you include your network cards and other interfaces with the world in that last portion. An interesting question is, if you want to understand a population of cells or a computer system, which of these stages would you like to be able to read the information at? They each tell you different things that could be more or less useful depending on the questions you want to ask. And still to date, we have very limited ability to read proteins, which would be analogous to like sampling live CPU registers off of a running machine. We have pretty well-developed abilities to read from disk and memory, DNA and RNA, though with some important caveats that single-cell sequencing helps to remove, which is why it's interesting. So just another take on this metaphor is that DNA is like the apps you have installed on your computer. There could be many of them. They're mostly not running at any given point. The apps are like genes. different machines with the same apps installed could be running different apps and present to the world very differently. They could, the same disk image, you could be operating as a web server or a graphic design workstation or an email video chat client or a development workstation. And those actual programs do send network requests and create audio and visual signals that actually interact with the world, and that's like proteins. So the important breakthrough here is to move from bulk sequencing to single-cell sequencing. All the genome sequencing that you've ever heard of for 10 or 20 years has been bulk sequencing. We take a population of cells, and we sample from the DNA and RNA across that population, but without any ability to resolve that to which specific cells those samples came from, which can be very useful for a lot of things. We've learned a lot from DNA bulk sequencing technologies as they've existed to date, but there are important questions that it doesn't help us answer, like when we really want to get into populations of cells where we know there are subgroups that are functioning as web servers or graphic design workstations, and they're interacting with each other in different ways, and just finding out that half of the cells are running Chrome or Terminal doesn't really tell us that finer structure that is important to get a deeper understanding of what types of cells there are and how they're interacting. So the ability to now sequence and resolve at single cell level instead of these bulk samples is essentially thanks to a clever chemistry trick where we pre-process the cells before putting them through the same bulk sequencing pipelines, which is great because we can reuse the existing billions of dollars of investment in genome sequencing machines. We still put them through these bulk sequencing pipelines, but we barcode them first. We have this like pre-processing step where the cells go through this very narrow tube that only one cell fits through at a time, and they get pinched into oil droplets with beads that are covered in barcodes. And a given bead has many copies of just one barcode. That's essentially a physical UUID. It's like a short sequence of RNA base pairs that is globally unique. There's just enough of them. There's A, C, G, or T, so it's 4 raised to the power of the length of the RNA sequence. So without going very long, you have a sequence that's unlikely to have existed anywhere in the world before. And you put the cells in individual droplets with these beads that are covered in barcodes, and the beads degrade the cell, and the barcodes hybridize to the ends of all of the RNA molecules in the cell. And now you can pool all the cells that are post-barcoding back together and push them through the bulk sequencing pipeline. But all the samples that you're getting of RNA molecules from cells have this barcode on the end of it that you know came from one of these beads that was only in a droplet with one specific cell. So if you group them in software according to their barcodes, you know that any reads with the same barcode all came from the same cell. If they have different barcodes, they were from different cells. So it essentially lets us join the reads back, or resolve the reads back to the individual cells that they came from. Not sure how clear that is, but it's not super important because after that, we cross the Rubicon from wet lab to dry lab, and we get all of our problems become software problems where we get what's called an expression matrix, which is essentially a giant sparse matrix of cells by genes that just says how many samples from each cell we got that showed that it was expressing each gene. And most of the entries are 0. Most cells are not expressing most genes, or they are in very vanishingly small quantities, so we don't end up sampling from it. And then we just have these matrices that we want to do various things with, like identify common patterns of genes that are being expressed in different cells, essentially cluster them together to identify cell types that exist in a way that's better than anything we were able to do previously. So this is an animation of the beads going through one of these droplet pinching pipelines, and then coming from down on top, in the real thing, there would be a cell which would get pinched into one of these droplets along with a bead. And most of the droplets end up empty. So many of the beads will go through and not have a cell in them, and cells might miss beads. But the important thing is if both channels in are dilute enough, you only end up with maximum 1 bead and 1 cell in a given droplet, and you filter out for those, and you just work with those downstream. So the Human Cell Atlas is this multi-year, multi-institute initiative to standardize around the data that comes off of these experiments, how we run the experiments in the first place, so that we can pool these data sets and generally do good science around this like exciting new kind of data that we can get access to. The ability to push cells through these pipelines has been exponentially increasing, which you can see on this chart. The largest public data set is about 1 million cells. And we expect even larger than that to come out whenever. It's kind of a big race among people that run these sequencing pipelines to try and do more and push more and more cells through these pipelines. Also, even given existing data sets that were done in separate experiments, like if they're both human cells, they have the same genes that are being measured, so you can, in principle, just concatenate the matrices and have a bigger data set with more cells that you can run a holistic analysis over, assuming that we've accounted for lots of biases that would otherwise creep in unless we were being organized in how different people run these experiments and stuff. So Human Cell Atlas, it's basically like the Human Genome Project, but 20 years later in response to a different sequencing breakthrough. Okay, so analyzing this data. As I said, we have these sparse matrices that are cells by genes. If it's human cells, then it's the same 30,000 or so genes every time. And the largest data set I mentioned was 1.3 million cells, so that's like 40 billion elements in the matrix. And most of them are 0. It's maybe like 1 billion non-zero elements. It's still it's of a size that you can just deal with on your laptop kind of today, but it's a bit unwieldy, and there's an emergent need to be able to use bigger and multiple machines to deal with this data. And especially if the data sets get larger, that will be a more pressing need. The kinds of analyses we do to this data are basically in 2 camps. There's like pre-processing stuff where we just want to, like we happen to get more samples from one cell versus another, and we want to normalize that out and just treat each cell like we got a distribution of what genes it's expressing. And then various flavors of clustering. We want to identify motifs of gene expression that are essentially types of cells that we then can understand what they do or how they interact with the other types better and stuff. So there's several pretty mature tools for dealing with this data that largely come out of institutions where they are generating the sequencing data, and the scientists themselves have built these tools that very effectively answer the questions that they have about the data. ScanPy is the most popular one in Python. There's a few in the R world. They all have pretty healthy open source ecosystems, and they all persist their data using HDF5 in one fashion or another, which I'll talk about in a second. So things that my group has wanted to help these tools do better is to process the data out of core, deal with data sets that are larger than fit in memory, often like leave the data sets remote somewhere, not have to download it to your laptop or your workstation to deal with it, and also operate on it in parallel. And it's hard to do these with HDF5, which has been the de facto standard format that these tools are using. So we kind of backed into having to think about how we store array data in this domain, and it has implications for other domains as well. HDF5 is a very esteemed standard way that a lot of scientific domains have stored data sets for decades. You can store heterogeneous data and the metadata associated with it in single large files on anything that looks like a file system, essentially, and transfer those large files atomically. The metadata stays co-located with the data that it describes, has a lot of good properties like that. It basically works by embedding a virtual file system inside of this single large file on your file system, like kind of like a zip archive or other things in that vein. You have this like nested directory structure where the directories are actually called groups, and you have files inside those groups that are data sets, which are multi-dimensional arrays of fixed types and whatever metadata you have that's associated with them. You can randomly access inside of these arrays and slice them in different ways, and it supports pluggable compression. An interesting distinction is that it's not really a file format, but more like a meta format, a meta file format. Like if you want to store data in HDF5, you still have to decide on a directory structure and names of the groups and the data sets and the shapes and the types that they will be inside your HDF5, so that somebody else's software that looks at one of your HDF5 files knows what to expect and can read them. It's kind of analogous to choosing a database to use, but then you still need to decide what tables you want to exist and what schemas those tables will have if you want other people to interact with your database. So it's kind of at that higher level of abstraction than a specific file format. Every different scientific domain has its own conventions around what files, directories and files they put inside their HDF5 files, and that's the second level that they standardize around. So we found it difficult to do what we want with HDF5 in a few ways. It's been around a long time. It has a lot. It's grown organically to an extreme degree. There's a very complicated spec. There's a lot of functionality that most people don't use, but that's in there. And there's only one bona fide implementation that I'm aware of that spec, which is this official C library that's maintained by the HDF5 group. And there's bindings for almost every language to work with HDF5 files, but they all essentially call this one C library that's a bit opaque, perhaps just as an outsider, but it's a bit daunting to try to make changes to it. It's not an obvious public issue tracker and things like this. It doesn't present as a regular open source project that I'm used to dealing with. In particular, it doesn't. The C library that everything uses it uses system calls to access local files only. There's no way to point it at cloud stores using, like Python and Java at least have pretty well established interfaces for just swapping in Google Cloud Storage or S3 or what have you inside some kind of interface that presents like a regular file system and lets you work with files that are either local or remote in these cloud stores. There's no real way to inject that into this C library. Like if you're using the Python bindings, you're kind of stuck. And it also, just by the design where you have these single large files, it doesn't play very nicely with parallel reading and writing. In general, it's very hard. If you have 1,000 workers and you're trying to write one giant file and have them each write 1,000th of it, number one, it's hard to do in software. You have to know exactly what the byte ranges are going to be ahead of time for each of these like random offsets, which is difficult if you're going to compress the data. And also, most file system-like things won't actually give you 1,000-way parallelism when you're doing that. It's like one disk head that's getting DOS attacked by your 1,000 workers, essentially. So that doesn't work well and kind of can't be made to work well as long as these things are supposed to be just one big file. And then parallel reading, it could work in principle. You can split these files and drop workers in at random offsets to read just disjoint shards of the data. But the general ecosystem for doing both of these is just not very well developed, and because it just hasn't been a priority for this project. These are kind of more recent concerns, I guess. And this project has been around a long time before we cared as much about that, I think. So in single cell and a few other domains, people were interested in finding something simpler and friendlier to remote storage and parallel processing. So Zarr is an interesting project that has a reference Python implementation and vibrant open source community around that. It basically does the same thing. You have these like directories and multi-dimensional array data sets that can be stored in nested directory structures. The arrays are chunked, and each chunk is its own file on an underlying file system, which plays nicely with the ways that parallel processing is typically done in the Hadoop ecosystem and elsewhere. So an example, simple like two-dimensional array is here on the top right, where this would be like the contents of one directory on your file system that has 4 of these chunk files that are each it's like 2 dimensions and 2 chunks along each axis, so 0, 0, 0, 1, et cetera. And a metadata file, z_array, that's just a JSON blob that tells you the size of the whole array, the size of the chunks, the data type being stored, which is like signed integers, and a pluggable compression algorithm. And the chunk files themselves are just those 10 by 10 chunks of integers in row major order, so just 100 4-byte integers run through zlib, and that's the file you have on disk. So it's much more approachable using official Zarr libraries and not. Spec is very simple. It doesn't have a lot of stuff bolted onto it yet. It's new. We'll get to bolt stuff on and make it harder to reason about over the years, but for now it does a few things very well, which is nice. And there's been a few implementations in other languages already, so the community around it is growing, and it's being somewhat officially adopted by the Human Cell Atlas for storing and processing this data. It also plays nicely with existing language bindings for talking to public clouds, for example, like in Python, the mutable mapping implementations of Google and Amazon just work for Zarr data that you access through the Python Zarr library. Something else relevant in this space of container formats is the common data model and its implementations called netcdf, which is an older project that essentially standardized around HDF5 and several other similar projects that all do a similar thing where they have these multi-dimensional arrays and some nested directory structure. They all have this in common, plus or minus some other auxiliary features. And so it represents that core that they all support. And it has at least a Java implementation that's from scratch of the bits of HDF5 that conform to this common data model. So most likely what's going to happen is Zarr will be one of the supported backends for this common data model. And can just exist in that community of things that are relatively portable from one to the next and across scientific domains, which is nice. So things that we've actually done to date, we've put Zarr support into ScanPy so that instead of just using HDF5 files, you can store and read and write data sets using Zarr single-cell sequencing data sets. So they work against cloud backends. There's definitely low-hanging optimizations to do, but I think the model basically works. We want to do similar things to the R tools, but haven't got around to it yet, and it's a little harder to see how to insert this abstraction in that ecosystem. We've done some work to improve the tooling around HDF5 itself as part of just doing technical diligence to see if we can make HDF5 work so we don't have to try to push a different format on people. We use the netcdf Java implementation of HDF5 and swapped the Java NIO file system interface into its I/O layer so that it can use existing Java NIO implementations of Google Cloud or S3 file systems so that we can read and write HDF5 files directly in these cloud stores, which to my knowledge is the only language where that exists. And we even put Spark bindings over the read path of that so we can drop, kind of like I described earlier, we can drop Spark workers in at random offsets in these arrays, even remotely in the cloud, even though it's HDF5 data, and have them basically look like a Hadoop input format that a lot of workers can read from in parallel. So some things you can make work there, but some other things are somewhat insurmountable. Tom built this library called Zappy that essentially presents an interface like NumPy arrays, but has pluggable distributed backends. So you can use Spark or Beam or Pywren, which essentially farms your array chunk computations out to AWS Lambdas, which is a really cool model that I'm excited about. And that's live in ScanPy, so you can process single cell data using any of these backends. And we ran some scaling benchmarks, and it does about what you would expect. Like you can throw more cores at it and have the runtime go down basically linearly, but subject to some upfront fixed cost of using these like heavier weight distributed tools, which is usually how that works. Dask is the original version of that trick where it's just like, look, it's like a NumPy array, but it'll distribute your computations on those arrays for you over a Kubernetes cluster. So we also support that as a backend in ScanPy, and that works well. And yeah, so things were planning to do to extend this is to benchmark on larger data sets. For now, we think we'll just take the existing public data sets and concatenate them to themselves a bunch of times to get synthetic larger data sets. But perhaps real sequencing pipelines will scale to these numbers soon. And we want to push this like parallel and cloud-friendly computation further in the single-cell pipelines. Today, it's just doing these like pre-processing steps that are a bit simpler, more just like map stages and less reduce and shuffle. And so things like clustering and indexing and searching are much more interesting algorithms to also lift into this distributed paradigm. And we want to expand all of this to the R ecosystem tools that are relevant here. And so yeah, that's where we are today. We're approaching this data that is processed today by a few different tools written in a few different languages, and we want them to be remote storage and parallel processing friendly across a few different parallelization engines. We have the initial scaling and distribution work is going about as well as we'd expect. And there's this library Zappy, which gives you arrays with pluggable distributed backends, which can be potentially relevant across a lot of different kinds of problems. So that's all I have. I'll take questions now if we have time.
What's the size of your data sets? Typically, for like 100,000 or whatever cells, what's the size?
Yeah, most of the data sets people are dealing with these days are smaller, let's say like 10,000 cells, and those would be anywhere from tens to hundreds of megabytes, and so it's really no problem to deal with them on a single node. The largest data set is like 1.3 million cells, and that's a couple gigabytes, I think, like compressed. And so when you actually uncompress it and iterate over it, it can be more unwieldy. Like people, to try to run these existing tools on that data set, people will use even hundreds of gigabytes of memory, and it hasn't really been optimized. It's kind of like no one's fully tackled that problem yet. A lot of times they'll just like filter out a bunch of the data before they try to process it, according to some hard cutoff is one way to avoid that problem. I don't know if that...
Okay, and so you said you optimized using Spark on storing the data, and at the same time you're using Zarr, but you're saying Zarr could be more I/O intensive. So which do you feel is better, Spark on SDF5 or Zarr on itself?
Yeah, so we can use Spark in a limited capacity for HDF5 data. It's very easy to use Spark on Zarr data, and it works much better like out of the box essentially if you point it at these Zarr directories where the chunks are already logical files. Like that's usually how Spark expects data to come in. It's like a directory that's a data set and it's been chunked into per chunk files inside that directory. So it looks just like that and it works almost for free in Spark. So yeah, does that make sense?
Hi, thank you so much. That was really interesting. Kind of the biologist, medical person question. It seems like there are a lot of potential applications with like cancer tumors and that sort of sequencing. Is this being used more and more within medical research?
Yeah, absolutely. Cancer is a big focus for this kind of data, particularly because like genome sequencing, if you're just getting the same information from all the cells, that works in like if all of your cells have the same germline DNA, but in a cancer setting, you want to know what's different from cell to cell actually, and that is important information. There are many companies that are aggressively pursuing drugs that work based on these principles, and it's a super active area of research and productionization. I'm not as plugged into that, but yeah. The answer is yes.
Got time for one more. So you mentioned Zappy. What are some differences between that and like Dask? You were talking about Dask data frames and.
Yeah, they're very similar. Dask has its own model for farming out the computations to workers that you register with it or that it is maintaining for you. As you know, it's like running a Kubernetes cluster on your behalf. And Spark and Beam and Pywren, the other backends that are supported by Zappy, are just different implementations of that same idea that have their own bells and whistles that may be better or worse, but it's kind of, there's an emergent need, I think, to abstract over these different ways to parallelize, to do this trick where it's like you want to pretend you just have this array and it's being parallelized somehow. There's a lot of choices that go into how that parallelization is implemented and what kinds of resources it's implemented on top of, et cetera, et cetera. Those tools are already, many tools are mature and have huge ecosystems that do that, and so we want to, if you for some reason know that you're using some part of this that works better on Spark, you can send it to a Spark backend, or if it works better on Dask, or you're more comfortable with these Kubernetes clusters that it is based on, you can use Dask for that. Yeah.
All right, well, thank Ryan again.
All right. Thank you.